This disease is a common lysosomal storage disease in which the patients have an autosomal recessive mutation. This mutation results in the accumulation of fatty substances in the cells and certain organs that cause malfunction. An autosomal recessive mutation is responsible for this disease. This means the child must receive the mutant allele from both parents in order to be affected. If both parents are carriers, there is a 1 in 4 chance that the child will not be a carrier or have the disease, a 1 in 2 chance that they will be a carrier, and a 1 in 4 chance that the child will have the disease.
Gaucher's disease is caused by a mutation in the glucosidase beta acid gene, or GBA for short, which is located on the long arm Q of chromosome 1 at position 21. Most of the GBA mutations responsible for Gaucher's disease occur through a single nucleotide polymorphism. This mutation results in the alteration of the enzyme beta-glucocerebrosidase, or beta-GC, by affecting its structure and preventing it from binding normally. There are over 250 other mutations affecting the beta-GC enzyme in Gaucher's disease. The beta-GC enzyme breaks down a fatty substance called glucocerebroside into glucose and simpler fat molecules called ceramides. Glucocerebroside is a type of lipid found in certain parts of the body as a result of the degradation of worn-out red and white blood cells.
The publication in Cell magazine by Dr. D. Rezik and his colleagues in 2007 found that beta-GC is tagged with the receptor protein LIMP2 for its transport to the lysosome. Beta-GC and LIMP2 bind together in the endoplasmic reticulum, and both proteins traverse through the Golgi and endocytic compartments en route to the lysosomes. The acidic lysosomal pH facilitates dissociation of beta-GC from LIMP2. Once the beta-GC and LIMP2 dissociates, SAPC identifies the active site of beta-GC enzyme and binds to it. This event activates the beta-GC enzyme, which then binds to glucocerebroside and breaks it down to glucose and ceramide. Mutation in GBA gene greatly reduces or eliminates the activity of beta-GC, and without this important enzyme, special cells called macrophages responsible for picking up and recycling glucocerebroside fill up with the undigested fat. These cells are referred to as Gaucher cells. Gaucher cells can accumulate in the liver, spleen, and bone marrow. The buildup of this fatty material can prevent these organs from functioning.
Philip Gaucher was the first to write about the description of the disease in a clinical case discovered in 1882 while Gaucher was still a medical student in France. But it wasn't until 1965 when Dr. Roscoe Brady and colleagues showed the primary defect in Gaucher's disease was a deficiency in glucocerebrosidase catalytic activity. Gaucher's disease is found mostly through the descendants of Ashkenazi Jews. There are 3 versions of the disease. Type 1, or the non-neuropathic version, is the most common type of Gaucher's disease in both the general population and the Ashkenazi Jewish population. It can occur in early childhood or adulthood, and its symptoms include bone disease, anemia, enlarged spleen and liver, bruising, and fatigue. There are 1 in 50,000 live births affected with type 1 Gaucher's disease in the general population, and 1 in 500 live births affected in the Ashkenazi Jewish population. Type 2, or acute infantile neuropathic Gaucher's disease, develops in infancy and generally leads to death by 2 years of age. Type 2 involves extensive and progressive brain damage and spleen and liver enlargement. Type 2 occurs in approximately 1 in 100,000 live births. And Type 3, or chronic neuropathic Gaucher's disease, progresses more slowly than Type 2 and can occur in early childhood or adulthood. Symptoms of this type include gradual brain damage, seizures, and enlarged spleen and liver.
While there is no cure for Gaucher disease, there are several treatments available to help alleviate its symptoms, which include enzyme replacement therapy, substrate reduction therapy, and bone marrow transplantation. These work best for the symptoms of type 1, but do not affect the brain involvement in types 2 and 3.